Okay, welcome back. This is lab 3. What we're going to be doing in this lab is looking at multi-lens systems. Now, where we left off last time, we took a lens and we collimated a divergent light source so that our sample, our ever-trusty ruler here, would be evenly illuminated so that when we imaged it with a lens and formed an image of it somewhere down the line, it had a nice, evenly illuminated field of view. Now in this lab, we're going to be complicating things slightly. We're going to be adding a second lens to our system. We're actually going to be making, in the first part at least, an infinity corrected system. So, as you learned in class, the infinity corrected system mandates that you put your objective lens one focal length away from your sample. So that's the first thing that we're going to be doing, is mounting our sample. Now, our objective lens, as you guys measured in previous lab, know that this is a 20 millimeter focal length lens. So just sort of quick little measurement that I'll be making sure that this is around 20 millimeters away from our sample. That looks about good. Now, we know that all the undiffracted light that does not interact with our sample is going to be focused to the back focal plane of our objective, right about there. So we know that if that's where the back focal plane is, then we need to mount our tube lens one focal length away from that back focal plane. So everything that is in focus here, all of the non-diffracted light, is perfectly out of focus in the front focal plane. So this is a new lens. We didn't measure this one, but I'll tell you the focal length now. It's 300 millimeters. So I'll take my little tape measure and we'll make sure that this is around 300 millimeters away from the back focal plane. Double checking. Looks good to me. Now, the image that should be formed is going to be another focal length away from this lens, and that's because our objective lens should be collimating our light, i.e. forming an image of its sample at an infinite distance away. And our tube lens takes that collimated light, basically something that is an infinite distance away from this, and refocuses it back down to an image at one focal length away. So, to quickly measure this, we're going to need to put our focal plane right about there. Looks good to me. Tighten that down. So the first thing that we'll do is just sort of sample our focal space and see how close we are to forming an image. All right, not too bad. All right, so you can see right off the bat, we have something of a blurry image being formed. You can see maybe a number, a couple gradations. So we're pretty close with these measurements in terms of 300 and 300. That's a very large distance, with a lot of room to play. What is probably misaligned or out of focus is our objective, right? Because you can see that even a slight adjustment, just a few millimeters, and my image is completely gone. So we'll need to move this very finely to make sure that our sample is exactly one focal length away from our lens, and you can see that we have a nice image of that 8 come into focus. Not only can you see the 8 and the gradations, but you can even see these little scratches, hopefully in the lens, or rather not the lens, but the ruler. So not only a highly magnified system, but also a high resolution one. So, well, I need to, let's try to get this 8 more centered into our field of view. So that's a little high. So let me try moving my sample down so that we move this 8 into focus. Oh. So as I move my sample down, my image moved up. So that should tell you something about whether this image is inverted or upright. So I suppose I need to go down to move this guy into focus. All right. So let me play a little bit more with this focusing, getting that 8 nice and sharp. All right. So that is about as close to in focus as I think I can get. What we're going to do now is use an empirical measurement to calculate what our magnification is. So I'm just going to move this back so where these little tick marks are visible. And we know that these tick marks represent 1 millimeter each. So this will make the math very easy, because what I'm going to do is make sure that we lock this sample plane down. And I'm just going to make, without moving the image very much, just a mark in the center of one of the ticks, and then a mark in the tick next to it. So I'll take these 2 marks and I will do a quick measurement. So to my eye that looks about 15 millimeters. So using the formula to calculate magnification of, we know that 1 millimeter in our object space is 15 millimeters in our image space, you should be able to calculate that our microscope has a magnification of 15x, 15 times. Now, that is the empirical way to calculate it. You could also have calculated that theoretically based on the focal lengths of these 2 lenses. Now, how does that number, 15x, relate to the focal length of this lens, which is 20 millimeters, and this lens, which is 300 millimeters? I'll give you a second to write that down. So what we're going to be doing now is messing with this sort of infinity corrected system so that we get conjugate image planes. So we're going to be mounting our sample not 1 focal length away from our objective, and we're going to be using this little iris to find where there are conjugate image planes. So somewhere in here, once I move this around, we're going to see an image of our sample. And then even further down the way, we're going to see another image of our sample. And that's going to require a little bit of maneuvering. So we will be back in just a second with a slightly different setup. All right, so what we're going to be doing now is looking at a setup with a multi-lens system where it's not infinity corrected, where we can look at conjugate image planes. So I have removed lens 2, and I have moved lens 1 now in such a way that we are now creating a real image. This is similar to what we did in the last lab. You can see that just with a single lens, we're creating a magnified image, right? And that's because our ruler is now mounted outside of 1 focal length of our first lens. All right, so the question is, let's find a way to put lens 2 in our system so that we form an image much further down the optical path. You can see I've actually had to mount this guy quite a bit farther away. So let's, oh, well, what you might even be able to see there is a image of our LED. Quite bright, but very much not at not at the right focal plane there. So you need to move this lens so that we form an image of our sample further down. Oh yeah, that's looking pretty good right there. Let's get that a little bit more in focus. Okay. So we have now found conjugate focal planes. we have a focal plane here that is showing these gradations. And then further down, we have another focal plane that is showing what looks like to be an even more magnified view of these gradations. So let's first try to describe this image. It's a little bit magnified, more so than we did in the last part of this lab. But now, we already sort of know intuitively that if I move my sample down, then my image at this intermediate focal plane is going to move up. So this should tell you something about this image. Is it inverted or upright relative to our sample? But now, let's look downfield again at these two markings that we've been able to fit in our field of view. If I move my sample down, my image also moves down. And if I move my sample up, my image moves up. So that should tell you something about the nature of this image. Is it upright or inverted? All right, so we already know where the conjugate focal plane is. It's right about here. And I have this nice iris that I'm going to place right at where I think this focal plane is. So let me just spend a second figuring out where it focuses the best. It's right about there. Just gonna hold my finger there, and we're gonna move my iris right there. So I've moved my iris there. It is fully open right now. Obviously, there's light coming through. Now I'm going to take a second and slowly close this iris and watch what happens to my image plane further down. What does it look like? Now if you want, pause the video and go back and look at what the iris looked like before I mounted it, because I can't easily take it off and show you here. But that shape there should be pretty familiar to you. It's actually the iris opening and closing. Those little triangles are the teeth. So that's fully open, that's fully closed. So this is what happens when you have mounted your iris in your conjugate image plane. And in fact, where I first estimated it to be is not the best focus, because I can move it over there and you can see that the edges of the teeth are a lot less sharp than when I moved it over here. So you can tell that there's an optimal space, an optimal plane. If I move it too far, it gets blurry. If I move it closer, I can get a nice sharp image of my teeth right there. You can see there's really narrow gaps and the teeth really come through when I have it in focus. If I move it even slightly out of focus, you can see they just blur together. This is a really useful way to determine conjugate image planes, is placing irises there. All right, so we know how an iris placed at this conjugate image plane affects the image downstream. Let's look what would happen if I moved my iris a lot closer to my objective, and then we'll play with this. Well, I just closed it. Now I'm going to open it again. We're not seeing the same thing that we did before. We're not seeing those teeth. That's interesting. And I have this pretty close to my objective right here. Oop, I bumped it a little bit. We'll get that back into focus. There. So as I close it, what do you notice about the image? It seems to be getting darker as I close it. So where I have mounted this now is probably right about at the back focal plane of my objective. So how does me narrowing this down affect the image? Let's try somewhere else. Let's see if I open my iris all the way up. You can see I just passed my intermediate focal plane there. Then I move it closer to my second lens. Now obviously I've narrowed down my field of view a little bit, but closing that, you can see we're still getting somewhat of an image of our teeth on there. They're just really blurred. And as I get closer to my intermediate focal plane, you can see sharper images of the teeth coming into focus. But if I go back, I'm going to remove this so I can get a little bit closer to my objective lens. If I go back over here, I'm just going to double check where my back focal plane is so that I know, putting it close to the right spot. It's right about there. My field of view hasn't changed when I keep this guy open or when I close it. You can see the same amount of the sample either way, but in the open case, it's quite a bit brighter than in the closed case. So this should tell you the answer to one of your questions on your sheet, that there is a location where the iris dims the intensity but the field of view remains the same. And that location is the back focal plane of our objective. You should be able to reason why that is based on your previous homeworks and your lectures. This is a useful way to dim the intensity of your image without putting any filters whatsoever, just blocking out different parts of the light. Okay? So that concludes Lab 3B. In Lab 4, we're going to be calibrating magnification in a much more precise way than simply markings on a piece of paper. We're going to be doing that with a digital microscope.